What if the very code that keeps your gut healthy begins to betray you as you age? That's exactly what research from the Leibniz Institute on Aging, FLI, in Germany, and the University of Turin in Italy has uncovered: a hidden mechanism that makes the aging intestine more prone to cancer. But here's where it gets fascinating. This vulnerability isn't just random wear and tear, it's the story of our cells' epigenetic memory slowly drifting off course. Over a lifetime, a subtle molecular shift, known as ACCA drift, builds up in the intestinal stem cells that regenerate our gut lining. These cells, which are usually adept at repairing and renewing tissue every few days, begin to accumulate changes in their epigenetic markers, the chemical tags on DNA that decide which genes remain active. When this drift intensifies, key genes essential for cell balance and repair become silenced through a process called DNA hypermethylation. The result? Pockets of aged, less functional tissue start forming, making the intestine more susceptible to colorectal cancer. The gut: fast renewal, slower protection. No other organ renews itself as rapidly as the gut. New cells arise constantly from specialized stem cells living deep within structures called intestinal crypts. However, as decades pass, these master cells undergo epigenetic alterations that stack up like miscopied notes in a genetic symphony. A research team led by Professor Francesco Neri revealed that this is not a random process, but follows a predictable pattern they named ACCA, aging and colon cancer-associated drift. This growing pattern of epigenetic noise becomes increasingly visible in older intestines, an unmistakable signature of cellular aging that appears again and again in colon cancer tissue. A patchwork of youth and decay. Here's the part most people miss: aging doesn't occur uniformly. Each intestinal crypt starts with a single stem cell, and when that founder cell picks up epigenetic errors, every descendant in that crypt carries the same flaws. Dr. Anna Krepelova from FLI compares this to aging patterns spreading like islands in a sea. Some regions remain youthful, while others lose rejuvenation capacity. Over time, the intestine becomes a mosaic of healthy and deteriorated areas, with the older crypts more likely to grow precancerous cells. Iron, the hidden key to genetic stability. Why does this drift even happen? One major culprit lies in iron metabolism. Older intestinal cells, it turns out, take in less iron, but leak more of it. This imbalance reduces the availability of iron, too, inside the cell nucleus, an essential cofactor needed by protective enzymes called TET, 1011 translocation enzymes. These enzymes normally clear away excessive DNA methylation marks, ensuring genes can switch on when needed. Without enough iron, TET enzymes lose efficiency, and methylation errors pile up, silencing critical genes responsible for repair and stability. As Dr. Krepelova explains, when cells can't hold on to enough iron, the protective systems that keep their DNA flexible begin to fail. Faulty chemical tags remain locked in place, and the cell's ability to correct them fades. The process snowballs. Less TET activity leads to more methylation, more silent genes, and deeper epigenetic drift. When inflammation and WNT signaling weaken. There's another twist to the story. The team found that low-grade, age-related inflammation in the gut acts as fuel for epigenetic drift. Inflammatory molecules interfere with iron balance and stress the cell's metabolism. Simultaneously, the WNT signaling pathway, a crucial system for keeping stem cells active, also weakens with age. The result is a dangerous synergy. Iron deficiency, inflammation, and disrupted signaling amplify one another, accelerating the pace of intestinal aging. As the researchers note, this means the gut's aging process may start earlier and advance faster than previously imagined. The hope: aging might be reversible. Despite its complexity, the study brings a note of optimism. Using lab-grown intestinal, organoids, miniature gut models made from real stem cells, scientists discovered that restoring iron levels, or reactivating WNT signaling could partially reverse epigenetic drift. These interventions reawaken TET enzymes, which then clear the excess methylation, restoring gene function. Epigenetic aging doesn't have to be a one-way street, says Dr. Krepelova. For the first time, we see it's possible to adjust the aging parameters encoded within the cell's molecular core. A new perspective on aging and cancer. This research, published in Nature Aging, Krepelova et al., 2025, reframes our understanding of how aging shapes cancer risk at the most fundamental level. It suggests that age-related decline isn't just about time, it's about biochemical drift that we might one day correct. But could resetting our cellular age also carry unintended consequences? Could reversing drift alter other processes we haven't yet understood? That's the debate waiting to unfold. Do you think epigenetic editing to slow intestinal aging is a promising breakthrough, or are we crossing into risky territory by meddling with the brain of our cells? Share your thoughts and join the discussion below. Source: Nature Aging, DOI 10.1038 S43587-025-01021X